All right. Well, thank you all for being here today. This is an exciting talk given our collaboration of VICC, CGC, and ClinGen Somatic Working Group on a recent manuscript on oncogenicity standards. We're really excited to have Dr. Wesley Goar here today to talk about some research that you've done on the adoption of these guidelines into different laboratory practices. So without further ado, I'll let you go ahead and get started. Before we do that, actually, I just want to reiterate, you probably noticed on the email that went out yesterday that we're purposefully leaving quite a bit of time for discussion. So please feel free to chime in, participate, share your anecdotal experiences, let's make this really interactive and fun. So thanks so much for being here. And without further ado, go ahead and get started.
Can you give me screen sharing permission, please? All right. Okay, I see it now. Let's see. All right, let's start. The Zoom bingo off, right? Can everybody see this?
Yes.
Yeah. All right, great. Okay. So yeah, I'm Wes. I'm going to talk to you guys about kind of some of our evaluation of the ClinGen CGC VICC oncogenicity guidelines and how it's supporting our pediatric variation classification workflows. So first off, I'm a senior bioinformatics scientist in the Wagner lab. Alex is the guy on the right here in the Hawaiian shirt with me. I have two beautiful daughters here that I have to show an obligatory picture of, and an amazing wife. And they're awesome and they're very supportive. A little bit of background about me. I have a PhD in computational genetics from the University of Iowa, where I studied rare inherited eye disease. I spent a lot of time rebuilding the whole genome and whole exome sequencing pipelines that were used in the Institute for Vision Research there. And I analyzed over 750 exomes while I was there for patients with like Bardet-Biedl syndrome, keratoconus, glaucoma, and some random samples of like nephrotic syndrome and familial non-medullary thyroid cancer. After that, I went and I managed the bioinformatics group at a cancer clinical reporting startup company, doing reports for patients over in China, where I worked closely with clinicians and providers to develop reports for them that were meaningful for both clinicians and patients. And then in March of 2023, Alex convinced me to come and join him at Nationwide Children's Hospital in Columbus, Ohio. And we work a lot on the standardization of genomic data and related concepts, specifically to enable the precise transmission of data between computer systems to allow for better analysis. And like Beth said at the beginning, I'm a really easygoing person and I really want this to be interactive and lively. So please feel free to interrupt at any time, ask questions. I want this to be a good conversation. So to give a little background about who we are, I work at the Institute for Genomic Medicine at Nationwide Children's Hospital. And our mission and vision really is to combine a robust clinical laboratory with genome scientists and clinical geneticists to optimize patient care and integrate molecular genomics into the mainstream pediatric patient diagnosis and treatment process. And at the IGM, we have an ongoing project known as the Molecular Characterization Initiative, or MCI for short. If you want to know more about that, Elaine Martis is on the call and you can bug her. This is a contract that we have with the NCI to do comprehensive genetic testing for kids that have cancer. And we received our first case almost a year ago, and we receive more every week. At the end of 2022, we had a total of 693 different patients that had been enrolled and consented and done over 2,000 different orders, including a bunch of methylation, fusion, and exomes. And we try to have obviously as quick of a turnaround time as we can, and that's something that we're always striving to get even faster. We had patients from 38 different states in the US, and we also had international patients from Canada, Australia, and New Zealand. And our variant scientists and clinical directors have to obviously analyze all of the results from these tests, along with any of the other tests that are coming through Nationwide Children's in general. And as you can see, this number kind of keeps continuing to ramp up. And that's probably fairly common for most people in clinical workflows as NGS continues to become more popular. And so, just like that, we know that the amount of genomics data and knowledge surrounding variants continues to actually expand greatly every year. And while we're able to actually perform assays like whole genome sequencing at an ever-increasing speed and reduce cost, the actual need to collect and evaluate all of that data surrounding the variants by a human creates a bottleneck in the speed of that analysis, as we're all probably intimately familiar with. And to be honest, variant scientists and clinical directors are being asked to evaluate more data than ever before as they're going through these tests and checking these results, while also trying to actually ensure that all of that data is relevant and meaningful and referring to the variant of interest. So one of the things that we're working on is trying to help our clinical teams through this variant analysis process and evidence collection specifically. And so to help alleviate the burden on our clinical team, while also, and this is important, and pseudo plug, developing an open source platform for pediatric data sharing, we decided to develop the variation categorizer, or VarCat for short. So this is all going to feed into where we're headed here, but one of the purposes of VarCat is really to severely reduce the manual searching and collation of variation data to help our clinical team spend less time with repetitive tasks by providing them with an interface that presents as much information as possible about a variant to them so they can focus more time analyzing the collected data and what it means for our cancer patients. So there's a couple services within the VICC that we employ, and if you want to learn more, please feel free to reach out to us. But one of those is the MetaKB, along with some of our internal normalizers. And through those, we can actually take data from different knowledge bases and aggregate and harmonize that data. And all that data is required for two of the clinical variation classification frameworks, the ClinGen CGC VICC Oncogenicity guidelines and the AMP-ASCO CAP guidelines. And this data is presented in a unified VarCat interface so that it can be used as evidence under these clinical variation classification frameworks. So users interact with the VarCat interface to assess a variant using these frameworks, while also being able to manually add and capture important evidence, such as like PMIDs from articles that are used to support conclusions or any pertinent notes that need to be retained. And the data from the assessed variant is stored in a standardized format and can be automatically reused or reanalyzed when the same variant is seen again, while also tracking and retaining any changes that have been made to that assessment. Where possible, we have also built an automated pre-calculated code assignment for the oncogenicity SOP and automated AMP tiering classifications on evidence from databases such as CIVIC or OncoKB for our clinical team to review and edit if needed. And any changes made to the automated classifications are captured for future analysis to determine future updates for VarCat. And we're actively expanding VarCat's capabilities and hope that it will continue to be a boon and really help our clinical team to reduce the variant analysis bottleneck. And again, like I plugged earlier, the other main goal of VarCat is to make this project open source for others to be able to use and promote pediatric data sharing so that we can all work together to help sick kids to overcome their cancer. And so one of those two frameworks that's feeding into VarCat is what we're going to talk about today, which is the ClinGen CGC VICC Oncogenicity guidelines. And I'm sure you're all familiar with them, but let's just kind of review. But these guidelines closely mirror the 2017 ACMG guidelines for variant pathogenicity. So you're all probably intimately familiar with this table. This oncogenicity guideline specification is based on a Bayesian framework and uses a scoring system to determine oncogenicity. The guidelines utilize data from many different sources, very similar to the ACMG guidelines for the most part, such as population data, functional data, predictive data, you have cancer hotspot data, and then you obviously have your computational evidence from like in silico predictors or splice prediction algorithms. And so just to briefly go over the codes so that we can start having conversations about these, you have your very strong category, or OVS1, which is simply put, you have a null variant within a bona fide tumor suppressor gene. It seems like it's probably fairly easy to figure out how to automate, right? Then you have your strong category, where OS1 is going to be your same amino acid change as a previously established oncogenic variant using this standard. This is something that we'll talk about a little bit. OS2 is going to be a well-established in vitro or in vivo functional study supportive of an oncogenic effect of that variant. And OS3 is, that variant is located in one of the hotspots in cancerhotspots.org with at least 50 samples with a somatic variant at the same amino acid position. And the same amino acid change count has at least, 10, has been seen in 10 samples at least. We also then have our moderate classification. So again, located in a critical and well-established part of a functional domain. So we're going to be looking at domains. You have, does the protein length change as a result of an in-frame deletion or insertion in a known oncogene or tumor suppressor gene, or a stop loss in a known tumor suppressor gene? You also have missense variant at an amino acid residue, where a different missense variant has been determined as being oncogenic, again, using this standard specifically. And then, further a moderate criteria for your cancer hotspot data. And then you have your supporting data, which is going to be, your 1 points here. And this is going to be like your in silico predictors are all pointing towards pathogenicity. You have OP2 is a somatic variant in a gene in a malignancy with a single genetic etiology, such as biallelic RB1 in a retinoblastoma. And then again, more cancer hotspot data. And then we get to our population data with absent from controls or really, really low frequency within NOMAD. And then the flip side, you have things that are going to push you towards benign, which are basically the flip of what we kind of just talked about. So where does this guideline set really fit into the process of your variant analysis? And it's right here in oncogenicity classification. So you have your identification of your variants, you annotate your variants, and then you try to determine which variants could be causing cancer, right? Could be leading to that. And then that would head into your AMP-ASCO CAP guidelines after, and then you have your clinical decision. So to give you an example of what we're trying to build right now, here's a quick screenshot of VarCat. And we have our variant and disease combo here with BRAF V600E, and we're checking this in terms of melanoma. And the first tab is going to be your oncogenicity classification. And you can see some of the different data that's required to actually determine the oncogenicity of a variant shown below, such as your in vitro or in vivo functional studies. And what we do here is we use Hypothesis, which is a way to annotate PDFs and websites to annotate specific links. And we can tag all of those and have direct links to all of whatever you're pointing to within there, along with being able to add your rationale and your description of why you're choosing that. We also, if you were to click on the little box right here, you'd have different codes potentially for downgrading or updating, while also being able to really, in terms of downgrading or upgrading your point values, we allow you to do that so that you can give less points or more points, and then you can explain why. If something maybe is a little bit unclear or muddy, we want to be able to capture that for future feedback potential. And then you can see we have things like, we call these just OncoKB cancer genes right now, as we're workshopping a title, but basically we went to OncoKB, took their list of all the 7 different resources that they use for classifying tumor suppressors or oncogenes, and we basically say you have to have at least 5 of those sources that agree that it's an oncogene or a tumor suppressor gene to make it into this list. Then you have cancer hotspot data, NOMAD, and data from DB and SFP. And we're filling this out. So basically we'll have all of the different codes that you'll have to go through to actually get your oncogenicity guidelines. And as you can see, anywhere where this purple computed value here, this is where we're automatically calculating these codes so that they're done for the variant scientists beforehand. And we're trying to do a lot of that, but automation is actually quite difficult when you start pulling from disparate resources that have very different data structures. And if you've ever tried like scraping the web through like journal articles and stuff to automate that, that's really challenging. And so some of these codes are quite hard, not only to automate, but frankly, this term causes me a lot of grief. This term well-established, right? And I understand why in the guidelines for both ACMG and for the oncogenicity guidelines, they use this term well-established and they leave it as ambiguous. But it's really important to actually start trying to define internally what that means and then come up with a consensus from the community, I think would be really wonderful to actually determine these. Because to be honest, what does well-established even mean? Right? Like Beth could say, well, I think well-established means this at Mayo, and Kathy and Elaine at Nationwide could say, well, I think well-established means this. And that might not be the same. And that's important to start really working on and capturing. And so I'm going to actually demonstrate a couple examples here for us why I think this is really important. Right? So as you remember, one of the codes is the variant has to be in a well-established and critical functional domain. Right? So if we're looking at the cosmic viewer here, this is PIK3R1, and let's say we have a variant here we're pointing to it purple, this is in the inter-SH2 domain, and we know that this variant actually causes problems with PIK3R1 binding to PIK3CA, which lets PIK3CA do whatever it wants, and that's not a good thing. So if we were looking at cosmic, we're like, yeah, this is great. Let's assign the code OP2. But if you're looking at UniProt and the protein domain track that they use, it's not there. And so unless you're familiar with this gene and the specific variant, you're going to immediately be like, okay, it's not there. Let's not assign this code. And then you're like, okay, well, let's look at cBioportal and whatever protein domain track that they're using. And again, it's missing, right? And so this is really important. Because even when we sit here and we say, well, what if we just take a bunch of different tracks and viewers and we say majority wins? Well, that would still not count here because 2 out of the 3 say no protein domain there, right? And so this is a specific challenge that we've had. And to be honest, we haven't fully teased out how we want to handle this yet because we want to show all of these protein domains so that we can make the educated choice. But what do you do when like the majority classifier is wrong? So that's a specific challenge. Next, and something that you're probably all really familiar with because of this being a long time with ACMG, is establishing guidelines for well-established in vitro and in vivo functional studies. So at Nationwide Children's, we obviously have our own list that we've come up with because of the ACMG guidelines on what we determine as, for rules such as applicability, validity, and validation of studies, right? And we have all of these different rules that we can apply. But again, do those actually match with what other institutions consider? And this is why we want to have these conversations. If you're pulling from something like Civic, they have a really nice evidence rating system where they've gone through and done a lot of curation on trying to help people make this choice. But again, not every database that you're going to be pulling from, especially if you're pulling from journal articles, they're not going to have any of that, right? And so trying to be able to figure out and come up with community guidelines on determining these things is really important. And then another pain point, at least for me particularly, right, is we have this single genetic etiology, right, where we say like, yes, I know with a surety that a biallelic inactivation of RB1 is going to cause retinoblastoma potentially. But variants in RB1 can also cause other cancers. And there's numerous clinical trials going on right now 4 RB1 variants that are causing not retinoblastoma, right? And with retinoblastoma being a fairly rare form of cancer, how often is this code actually going to be applied? And should we consider figuring out some kind of threshold or frequency for being able to say, like, look, the variant in this gene has been implicated or seen in this cancer X percentage of time? And this is another thing that we want to talk about and come to some community, guidance with. And because of the mirroring of these guidelines to ACMG, I think we all understand that this code works a lot better for rare inherited disease than maybe for cancer. And then pediatric versus adult data. And I think all of us who work on pediatric data know that most knowledge bases are very heavy adult-centric data. And so not only do we want to find and harmonize as much pediatric data as we can, but we also want to promote pediatric data sharing, which is part of the reason why we're building VarCat, is we want this to be an open source platform that other people can use, and we can share variant analysis from pediatric cases with one another. And this all leads into this process that Alex likes to call the virtuous cycle, right? Which is where we can take these guidelines and we can put those into a framework, our VarCat interface, for instance, and we can have people start using that. And they can go through all of the codes and they can write down the rationale for whenever they downgrade or upgrade, or they choose to or not to apply certain codes. And we can capture all of those things and actually collect data, right? Like, how often is this code applied? How often is this code not applied? And in what types of cases, right? We can take all of that data and then we can push that back to the community, present that, and have good, strong discussions about how we can update and further improve the guidelines so that we can, again, back to the main point, help kids overcome their cancer, right? Come up with the best data possible. And so this is what I want to talk about for the rest of the conversation, really, after I make a quick plug for the hackathon that we are hosting in tandem with Civic and ClinGen Somatic at CGC. If you're coming to CGC, please join us at the hackathon. This specific topic is one of the issues that I have specifically proposed to talk about at the hackathon, because it's not just for people who are very programmatic heavy. We're also doing, curation jamboree stuff. I have 2 QR codes here. The one on the top takes you to the VICC landing page that shows you this. The one on the bottom is actually the GitHub issue page where you can go through and submit your own issues that you would like to talk about at the hackathon. So please join us. It's the day before CGC starts, and we're really hoping that it's going to be an awesome hackathon. All right, now I have my acknowledgements, and then I want to have some nice discussion. And this QR code at the top left, there's a Google form that you can actually go to, and you can submit your own feedback and thoughts on how the guidelines have been maybe applied at, or you're trying to apply it, or thoughts in general about this process. So then again, we can keep spurring and talking with the community about application of these guidelines. And then obviously, thank you to everyone on this list. They're all great and very supportive. And now let's have a great conversation.
Thanks so much. Feel free to jump in, anybody who has comments or questions or experience in trying to adopt these guidelines. Oh, go right ahead.
Hello.
Oh, hi, Paulo. Yep.
Very nice presentation. This, have you?
Paulo, you're
breaking up a little bit, Paulo.
Yeah, Paulo, you're breaking up a lot. I can't.
Criteria where at an amino acid residue, but can you hear me?
You were breaking up a lot. I didn't catch anything besides hello, which hello back.
Oh, I'm sorry. Can you hear me now?
Yeah, you seem to be better
that's
now.
much better.
Good. So I'm just curious if you have tried to automate the OM3 criteria, where you have a variant in the same amino acid, but the distance must be higher than previous pathogenic. Is that automated?
So we have part of that code automated. We haven't added like any Grantham scores or anything yet to do that calculation. Right now, we're simply focused on, because we're trying to see, like, has this been seen before? A lot of effort that's gone into this tool has gone into historical tracking, and there's been a lot of effort to build out so that we can do evidence tracking from different groups. Like, we already have that built in so that we could drop this to someone else and be able to have data sharing. And so a lot of the effort there has been just, how do we store the data in a way that we can, one, identify that it's been seen before at the same location, and then we'll work on adding things like, your Grantham scores or any other different coefficients that would be routinely used.
Yeah, that will be useful, because this is a kind of a tricky point. I mean, this distance, and we very often have variants in the same amino acid as a previous pathogenic variant. Okay, good.
Yeah, and not only that, right? There's the specific line in the code that says classified as oncogenic using this standard, and nobody's published that yet. So it's like, all right, like, so how do we, do this? And so that's why we've worked a lot on our own internal evidence, right? So then we can, hopefully other people can use this, and then we can all share. So if we drop this over to you, Paolo, you could say, oh yeah, NCH has seen this already. Great. Like, we know someone else has already classified this as oncogenic.
So I see that.
And another tricky part of this criteria is if we should use only oncogenic, or if a variant has been classified as likely oncogenic before.
Exactly.
Right? We meet that a lot. I mean, that situation. So you have a previous one that was assigned as likely oncogenic, and then you find in the same amino acid. It's not very clear. I mean, a lot to discuss.
Yeah, and so the way that we've thought about that right now, Paolo, is we actually, if it's likely oncogenic, we are trending towards actually downgrading it a point, like a point scale down, just because because we want this on this Bayesian framework. We know that if it's oncogenic, that's 99% probably true, right? Versus likely oncogenic is a larger gap. And so we've thought about, okay, well, if it's likely oncogenic, let's downgrade it a point so that we can be a little bit more stringent.
Good. Congratulations for the work.
Thank you. That was really good points. I see that we have a couple hands raised. So I think, Karen, you had your hand raised first.
Hi. Thanks, Wes. That was really great. I was wondering, does VarCat only contain confirmed somatic variants, or does it also capture variants where it's not confirmed, whether it's germline versus somatic, or like known germline cancer predisposition variants? Yeah, so short answer is VarCat can be run on anything, right? So you can run it on your variant disease combination. The way that we're going to be applying it is going to be coming out of our paired sequencing generally with your somatic, and then another part of the VarHouse team here is working on the germline tool that's going to use the same framework and will be reskinned to do ACMG guidelines. But you can run it on anything if you really wanted to.
And we have another question from Brendan.
Hello, Wes, that was an amazing talk. Thank you so much. That was super informative. I love how you've been thinking about this and very, very clearly. My question was, when my initial read of the guidelines was, I totally agree that like well-established and such is ambiguous, but in terms of the categories, it seems like the functional evidence is probably the hardest to crack down and to get firm from a kind of a field point of view. I know there's been some really heroic efforts with something like the BRCA exchange and p53 portal, I think it's called, to kind of just test individual nucleotides. And so I'm just wondering if you can kind of speculate and talk about how we kind of get a ground truth there. In interpretation, we always get asked that. And it's like, well, the ground truth is always changing because papers are always coming out. That's especially the case preclinical. But it is it is a fire hose. So I'm just wondering, like, for you to talk about that some, and then also, like, do you think that's maybe super hard? Does it actually matter? Like, is the effort maybe not, is the lemon worth a squeeze with the preclinical stuff? So you can take that question however you want.
Yeah, I think this is a really great point. Something that we're really excited about, specifically Alex and I, is our work with the MAVE Alliance, the Multiplex Assay of Variant Effect group. And at CCG this year, where there's actually going to be a workshop talking about how we could potentially use MAVE data within a clinical setting for this purpose, right? Of being able to say, like, look, we've hammered all of these different variants and we've seen, an effect. Again, this is hard, right, Brendan, because just because it does something like on the dish, does it have like a real biological effect, right? This is always really challenging, like within your complex human body. But at least it provides you some, usable data for that. And I think this is an ongoing conversation that we would really love to talk about in general more, not only like the use of MAVES, but in general, like how trustworthy is the paper that you're like, and the study that you're pulling from, right? How do you classify that, right? How do you categorize all of those different points of quote unquote like applicability or validation or validity? And it would be really awesome if as a community we could come up with some firm guidance so that we know as we start sharing more and more data with each other that yes, you're just as stringent as I am, right? And I have the exact same confidence level in your data as in my data.
Yeah, no, totally. Oh, go ahead, Beth, sorry.
Oh yeah, no, that's okay. Yeah, I was going to agree and just say, with that being said, cancer is so nuanced. I mean, you could be looking at a lung adenocarcinoma, and we know a lot about lung adenocarcinoma, and then the next minute you could be looking at a rare tumor type, and the amount of evidence that's going to be supporting variants, oncogenicity in those two tumor types is going to be quite a bit different. So, like, navigating what well-established means in different cancer contexts, I feel is really challenging. So kudos to giving it a whirl and trying to figure it out.
Yeah, and obviously like ClinGen Somatic is doing heroic efforts on specific genes, right? Which is really wonderful. But that's going to take a lot of time to get through. Whereas this is a general framework, right, to be applicable to, everything. And we don't necessarily have the time to be able to go into the level of nuance that they do, because, it takes them a couple years to really get that end product and have that like firmness. And their data is awesome. But there's just not enough people to contribute to that right now. And so I think this is why we really just want to keep spurring, let's get the community to work together on, shoring some of these things up so that we can all work together to help, especially the pediatric group that has less data than, all of the adult data.
I wonder if something like VarCat would be able at some point to like automate a ClinVar submission or something like that, and then like you would reach all the people that go to something like ClinVar. But yeah, really interesting stuff. Cool.
As it so happens, we've got Melissa Landrum on the call. And for those of you that are unfamiliar, Melissa runs the ClinVar resource. And one of the things Melissa and team have been working on is creating like a somatic classification framework within ClinVar so that you can submit these types of data. And it might be an opportunity for Melissa to speak a little bit to those plans in progress.
Yes, thank you, Alex. We are working on adding, we consider it improved support for somatic variants. We have always kind of thought that ClinVar should be supporting somatic variants, but we sort of learned that the communities didn't feel that way. So what we're doing to make it very clear is we're adding basically 2 fields for somatic classification. One is clinical impact, that's using the AMP ASCO CAP tiers. And one is oncogenicity, so using the oncogenicity terms you guys are talking about today. So we are working on that now. We think we'll have it available this summer, initially for file submissions, but we will be adding support through our submission API. I think Beth's suggestion just now was, being able to sort of automate submissions from other software to do ClinVar submissions. So that would be coming later on too. And I'd be happy if this group wants like an overview. We have some mock-ups of what the web displays will look like for somatic variants, the submission template, our XML. I'd be happy to come back another day and kind of present what we're planning to do and get some feedback.
Yeah, we'd love that. Yeah, we'll be in touch.
Excellent. Thanks.
And then also to kind of build on that, Wes, I have a question for you. If do you find that people who are adopting the oncogenicity standards are reporting an oncogenicity classification alongside an AMP ASCO CAP tier, or is it more kind of assay specific?
This is a ongoing hot topic with our clinical team, and something that we've talked a lot about. And there's been no firm established results from the group as we continue to, have really what we're starting to think about now is we're at the point now where VarCat is about to be deployed in like beta phase with our group. So we can start actually collecting data, because you can keep coming up with these scenarios of like, okay, well, what if the oncogenicity guidelines classify it as a VUS, but we know that this is a tier 1 level A variant, right, in the AMP ASCO CAP guidelines. You can have that hypothetical scenario, but what if that actually never happens? And so trying to actually tease out some of the initial data that comes through so that we can use this, right? And so, Kathy, you can absolutely override me if you disagree with this, but I think our current thought process as we're moving forward is we probably will not be quote unquote reporting the oncogenicity guidelines. We'll be reporting the AMP ASCO CAP tiering, but we're going to be using the oncogenicity guidelines for variant prioritization analysis.
Yes, Wes, I would completely agree with you. I think performing that initial oncogenicity framework up front helps us really to establish a lot of the evidence that ultimately does feed into the tiering strategy as well. But it's a way to sort of collate that together, have it all documented, and then that will ultimately help inform the tiering, which is what shows up on the clinical report in terms of diagnostic, prognostic, or therapeutic significance.
Great, thanks. Oh, that's good. I see Alex has his hand raised, so go ahead.
Thanks. Yeah, one of the first of all, that was a fantastic presentation. Great work, Wes. Second, a couple of the comments that came up in the Q&A here had me thinking about something else that we've discussed that wasn't part of the material we presented today, which is kind of how we use mutation recurrence data from resources other than cancer hotspots. So the current codes, OS3, OM3, OP3, they're kind of focused on these like count-based thresholds for amino acid changes that exist in cancer hotspots. And I know that we've had some discussions with the clinical team about kind of how we use cancer hotspots as well as other resources relevant to pediatric cancers in order to, kind of get those codes applied. I wondered if you had any thoughts or comments on ways that we can scale and generalize these codes to work with other resources beyond cancer hotspots. open question for the community, not just Wes.
So I know like some groups will look at different databases like Cosmic or even CBio Portal, and then you can say, this amino acid residue has, been seen in 20 different samples. Does that substantiate a like pseudo hotspot, even if it's not in cancer hotspots? We know that it's established or that it has been seen or verified somatic. So the question is, yes, that holds some weight, but how much weight does that hold versus like a cancer hotspot that's been identified as a cancer hotspot?
Yeah, if I could jump in, I think that's really challenging because the cancer hotspots is still a relatively small database. And so there are a lot of genes that are involved in cancer that aren't in it. And just the number of reports are so much smaller. So I think a combination with Cosmic, and Cosmic has its own issues because it can be dirty and there are repeated there are duplicates, it's hard to interpret that as well. So I think a combined approach is warranted for that rule.
Yeah, I think that probably the use of other databases
I'm on pins and needles here.
on Cosmic for can you guys hear me?
You were cutting out quite a bit. I think maybe you've locked in again.
I'm sorry. Sorry about that, guys. My internet connection is not good today. Can you hear me now better?
Yeah.
Yes.
Well, I was just saying that probably the use of other databases, not other than cancer hotspots, should be specific for each gene, I think. So for some genes, Cosmic with some very specific criteria might be useful. We are studying that in some VSEPs, like FGFR3, trying to define a cutoff where X number of appearances in Cosmic will be it's in the classification, but it's very tricky, as was mentioned. The use of Cosmic is because it's dirty. We have a lot of benign variants in there, and so in my opinion should be gene specific. But I have another question to pose to the community in the follow-up of what this do we have any evidence to think that a variant in the context of a pediatric cancer should be somehow treated different from any other variant in cancer? I mean, is there any specificity other than the frequency that it happens, because that varies a lot, right? We have some variants that are way more common in pediatric cancer and others in adult cancer. Thought about should we use any different weight or anything if the variant shows up in a pediatric cancer or in adult cancer? Because to my understand, I think probably we should not, right? But I would like to hear other people. What do you guys think? I mean, basically, should we curate the variant different in some way if it's a pediatric cancer or an adult cancer?
Paulo, I would agree with you. I think the challenge is at times pediatric cancers have been underrepresented. And so I think that was some of the thought process surrounding this. What is the best way to aggregate data and ensure that You know, the oncogenicity guidelines are supportive broadly of a variety of different cancer types.
I mean, I would say if it's the same variant that does occur in pediatric and adult cancers, probably the same principles will apply. The problem is that pediatric cancers will have totally different variants in completely different genes where you struggle because of what Kathy just said, the representation and lack of information. So this for the same variants, probably not a huge difference, but then there are so many that will be unique and in different genes that in that sense, there is difference.
Yeah, I would agree with that and say that the clinical implications are going to differ from disease to disease, and that's where the AMP guidelines are so important with the levels of evidence. But the oncogenicity and ACMG classifications are critical for the, you know, the biochemical, what is this variant doing? And I don't think that changes as much from, or at all from cancer type to cancer type.
And there is also just this principle of whether a gene makes sense for the tumor type, which we kind of get into calculation every time we look at a variant. So I would look at the gene and does it make sense for the cancer in front of me? And in that sense, you will have difference in pediatric cancers as well.
Yeah.
So the context come into play that way.
That's a very tricky point, right? Because
You're breaking up again, Polo.
yeah. But I mean, in the pre-rating of, sorry about that. Can you hear me now?
Yes, we can hear you now.
Yeah, sorry. Well, I was just mentioning that when we are curating a variant, many times we may not know, right, what's the relevance of that variant for that kind of cancer. I mean, it may not be a frequent target, but I mean, I think it's hard to judge, right? When at least our approach is whatever variant shows up in any tumor type, we classify that using the oncogenicity score. And if it's oncogenic or likely oncogenic, we report. Then the other step will be to try to understand what's the clinical relevance. But it's very hard to know, like, we have variants in genes that shows up in 1% of specific tumor types, like APC variants in, I don't know, melanoma, right? Sometimes we have a 1%, but I think we have no criteria to assign, is this oncogenic in this, is this contributing to oncogenicity in this tumor type or not? I mean, this is tricky, right? I mean, to make these kind of decisions.
Yep, it is. We don't need VarCat for BRAF V600E. We need it for these hard ones.
Yeah.
And they love it.
To that end, one question I had then, especially regarding what do we do about variants and genes that aren't represented in cancer hotspots, is the scope of what you're maybe thinking about for a VarCat study to do almost like an A/B test of take 50 molecular pathologists, present to them some number of variants that are not in cancer hotspots, and then maybe like, here's the information in COSMIC, here's some without the information in COSMIC. What are the sentiments that these pathologists are expressing? Does it actually add value to make a call? Or is the kind of goals of this resource you're creating to kind of just, or not just, do you want to kind of take the approach of show as much information as possible and kind of let the community decide, or kind of do these A/B tests to figure out if values, to see if there's being value added? Do you have any thoughts on that?
I think really like it's a little bit of both, right? We want to provide as much information as the community wants, right? So right now, specifically, it's really been with our clinical team, what they're asking for, right? And then once this goes out to open source release, people will be able to go back and say like, oh, I would really love this data to present it if you can get it this data, right? And then the whole time, the point is we allow you to add your own like evidence or data into it also, so not just what's presented, so that that can be captured, right? And then as you get people utilizing it, you can start seeing trends of, okay, like this is where cancer hotspots has fallen short. We don't have data from that, but people have cited or sourced this data, right? And they've used that, and being able to tease that out, I think will be a really valuable thing. So then we can go back, and again, we just have this cycle of improvement, hopefully, that happens as we all work together to, you know, come up with the best resources and guidelines in general for handling some of these tricky situations.
Yeah, no, absolutely. Similarly love the process of the virtuous cycle and stuff and such. Has like, I'm not as familiar, like ClinVar and such also kind of dealt with this, where it's almost the struggle between information overload for the molecular pathologist, right, and careful curation, which is one of the things what makes the guidelines so great.
Yeah, I think that's kind of the role of these tools like VarCat, is to help translate between, you know, active practice, we're trying to get the job done and we want to do it in a way that doesn't overwhelm us, or in fact reduces our burden, right? And the idea that these tools also apply, like use standardized interfaces and data exchange formats to basically allow for the transmission of these data to other resources, as well as to allow for the integration of these data and downstream efforts, so that you can reuse it and you can combine it with other pieces of evidence in a way that's not just your final classification, but, you know, it's all the underlying bits and components that comprise it, so that you can then create like updated classifications as more evidence is available down the road.
Great, awesome. I'm really enjoying this discussion. I see that we only have about 5 minutes left. So I'm wondering if you would have any advice for people who would want to start adopting these newer standards and guidelines and start automating them. I know that we know a little bit more about VarCat now, and that's freely publicly available. But what are some tips that you would give somebody maybe in a small lab that's trying to adopt these guidelines into their own practice?
Yeah, I mean, I think, and you know, Kathy will probably vehemently agree with this. I think before you start doing it, making sure you establish how you're going to handle some of the ambiguity is really important. You know, especially in clinical scope, right? You want to lock it in before you start doing it, and then, you know, stick with it. And so we've had a couple of practice sessions where we've taken some challenging variants and tried to run it through as we've been developing it, just manually going through it. And there's been just like the scope of actually how different scientists have classified a variant and their thoughts on whether they're following it stringently versus best judgment kind of thing has been, there's actually some quite change, right? If you're like, oh yeah, but from my other understanding, I can say like, maybe I'm going to bounce it this way a little bit or this way, versus if you're following it to the letter. And so I think, especially in a clinical setting, you really have to sit down and just be cognizant of locking in how you're going to handle the ambiguity before you start like committing to do it. And again, right now, we're really thinking about using this as kind of a prioritization scheme, right? Where we can go through and say like, okay, here's the variants that classify as oncogenic. We're going to look at these first. But just in general, I think really having meaningful discussions with your team about the areas that are ambiguous and locking those down is super critical.
Awesome, great advice.
Hey Wes, really great talk. I just have one quick question. For the VarCat, the workflow you presented, could you elaborate the source knowledge base your team have used? You mentioned, the ClinVar, OncoKB. What other source knowledge base you have used for that workflow?
Yeah, so I mean, for the oncogenicity guidelines, there's a bunch of different knowledge bases, right, that feed into that, Jeff. And because you're part of the MediCAB project, you know that we utilize a lot of our normalization services to handle some of this data. You know, what I didn't show on VarCat is we also have AMP ASCO CAP in there, right? And so we'll be using information from CIVIC, Molecular Oncology Almanac, OncoKB, and then others as we either ascertain permission or get those and harmonize that data under the hood. And a lot of that harmonization is done through MediCAB and our normalization services through the VICC, if that answers your question.
Yes, yeah, thanks, Wes.
Well, thank you all so much. I really enjoyed this conversation, and I really look forward to continuing to hear more from you guys so we can work together to overcome some of these challenging, sticky issues.
Awesome. Yes. Thank you so much for being here, presenting, and coming up with some really great topics for discussion. Be really interesting. It'd be really fun to have you back here in like a year or so and see how the landscape has changed over that time.
Yeah, that'd be awesome.
Really, really cool. Yeah. So thank you so much for being here. And I see we're kind of at the top of the hour. So thanks everybody for contributing to the discussion. And this will be on the YouTube channel probably by the end of the day. And then we can, you can direct people there. The QR codes, we can probably send those around again. And yeah, if you have any other questions, you can contact Wes or any of the VICC leads and we'll triage those.
Thank you so much.
Thank you. Have
Thank
a good day.
you.
Bye.
Thank you. Bye.